Tanse, mio Kizakanesik. It is my very great pleasure today to introduce you to my friend and colleague, Dr. Warren Cardinal-McTeague. First, though, I'd like to mention that I am joining you today from the homeland of the Lekwungen-speaking peoples, the Songhees, Esquimalt, and Wasanich nations. Dr. Cardinal-McTeague is joining us from unceded territory of the Algonquin Nation. Most of you are joining us from somewhere along the shore of Lake Michigan or the greater Chicagoland area. And at the Field Museum, we recognize that we occupy lands that have been inhabited by First Peoples for millennia. So these include the Ho-Chunk, Missouria, Menominee, Mishkwekika, I'm going to butcher this, Ashakwaki, the Sauk, Miyamiki, Wayatnoki, and Pionkasaki, Kikakapoy, Anishinaabeg, Odawak, and Potawatomi, or the Potawatomi peoples, as well as the Illinois Confederacy. Many of these nations have been subjected to genocidal actions as well as systemically racist practices of displacement and forced assimilation. Today, Chicago is home to thousands of Indigenous peoples from across the Americas. At the Field Museum, we recognize that our institute has been historically complicit in harms, both through extractive colonial practices and as a cultural touchstone of settler colonialism. We intend to navigate a path forward into justice and good relations. And in that context, we are delighted to reinitiate the Armor Seminar Series for Native American Heritage Month and to welcome Dr. Cardinal-McTeague. Warren is a citizen of the Métis Nation of Alberta and is Nahio, Métis, and Irish American by descent, and maintains community connections in his hometowns of Fort McMurray and Lac La Biche, Alberta, in Treaty 8 and Treaty 6 territories, respectively. Warren earned his doctoral degree at the University of Ottawa and the Canadian Museum of Nature, and was a pre-doctoral fellow at the Smithsonian Institute, and subsequently held an NSERC postdoctoral fellowship at the Institut de Recherche en Biologie Végétale at the University of Montreal, Quebec. He is currently a research scientist with Agriculture and Agri-Food Canada. Warren has a deep connection to natural history collections and collecting around the world. He's engaged in botanical fieldwork across the planet, from the highlands of Madagascar to the tropics of Brazil. In addition to his systematic and biogeographic work on flowering plants, Warren is bringing an Indigenous lens to plant sciences. And so we are delighted to welcome him to the Armor Seminar Series. Warren, I'm going to let you take it away.
Thank you so much, Az. So I cannot see the audience, but I just want to say tanse and hello to everyone. I know I might know some of you, and maybe even some of you I haven't seen in a while. So if you are there, just know I see you in spirit. And I'm going to go ahead and get started with my presentation. And as it was mentioned, I'm going to talk to you today about my sort of research story and how it sort of evolved from sort of a purely Western scientific endeavor to one that bridges in Indigenous perspectives. And so I just want to draw your attention to the beadwork illustrated on the right. And this is work that is done by my little grandma, or my great-grandmother, Christine McDonald. And you'll see I interspersed Indigenous art throughout this presentation. And it was intentional to draw attention to the scientific knowledge that's embedded in art and how art is a very effective form of communication. And so I hope you gather some of the sort of unspoken messages through the art that's presented today. So let me see, change the slide. There we go. So again, thank you so much for this invitation to the coordinators of the seminar series and for everyone who's in attendance. I want to start off by recognizing that it is Native American Heritage Month and that it's a real honor to be here speaking with you today on some things that are very important to me. So in addition to that, I will also reiterate some of what Dr. Klimiak has mentioned that we, I'll just mention it for Chicago area. So I recognize that if this was in person, we would be presenting on the traditional and unceded territories of these many nations that call the Chicago area their ancestral and current homelands, including the Ho-Chunk, the Kickapoo, Menominee, Meskwaki, Miami, Peoria, Potawatomi, and the Sauk. And there's been a lot of discussion on land acknowledgements and how they become performative, but I think it's worth reiterating that they served a very strong purpose initially, which was to unsettle those settler spaces. So it really did a real job at the beginning to really shock people into awareness that yes, this is Indigenous land and that this comes with some change in how we've engaged with it. So what it's really meant to do is start a conversation. And what we need to do now is sort of dig deeper beyond just that acknowledgement. And so what I like to do is illustrate what it means to me. Sort of these 3 bullet points here at the bottom is sort of to learn about whose land I'm either on or visiting, similar to what we did just now, sort of recognizing these different nations. It also involves respecting their authority and autonomy over the lands that they govern, as well as to support them in their time of need. And I usually dedicate a bit more time to this last part to illustrate how, not easy, but like some people find a hard time, like, oh, how can I really support them? I don't know them. But I think the news and just opening your eyes to media will really highlight the lots of the issues that Indigenous peoples are calling for help from. So one of the brief examples I'll mention is something nearby to where you all are at, which is the Minnesota Water Protectors and the Line 3 pipeline expansion. And these are Indigenous communities exercising their rights to protect the land, protect the water, and protect both the livelihood of their community and the plants and animals and non-human kin that live within it. So it's really quite easy. This could just be slides and slides of current event asks for help. So support them however you wish. Can be in person, can be financially, can be through raising awareness. These are just some examples. So Alice had already given me such an amazing introduction, so I'm going to breeze through some of this. But I will mention that it is like a very important Indigenous protocol to introduce who you are, where you're from. It helps to sort of break down barriers. So maybe if some of you are coming to the chat afterwards, you already know a little bit about who I am, where I'm from, makes me a bit more approachable. It also sort of helps hold me accountable to myself, my family, and my community. So I'll just briefly say in Tansei, Nea Muswa, Nea Apitogesan, E Nehiao, Kaksino Masuyan Segaguna, and Wapogonia, A Odawa. So I just spoke to you in Plains Cree or Nehiyawewin, which is the language my family speaks. And I don't speak too much of it now, but we try to speak whatever we can. And I just said to you, my name is Muswa, and that I'm Métis and Cree, and that I study or I learn myself plants and flowers in Ottawa. That's sort of a way of expressing sort of who I am and what I do. As is sort of mentioned, I am of mixed ancestry. So the cardinal side of my family is Métis and Cree, and the McTeague side of my family is Irish, German, American, who immigrated to Canada from the United States. And I'm always reminded, this is sort of a new slide I've added, because sometimes it's sort of breeze through focusing on my Métis and Cree side. But I was reminded by a colleague of mine who said, Samantha Davis, she said, will you honor all of your grandmothers? Like, will you honor all of your ancestors, or will you leave some out? And I thought about the responsibility that means. If my family was here and I were to just leave them out, they'd be really offended. And so I think it's really important to honor all of your ancestors and all of your ancestors and be able to. And in addition, I'm also proudly gay, queer, and two-spirit. And so here's a photo of my partner, Francois René, and our adorable little puppy named Popcorn. And basically, I also volunteer this in addition to my Métis and Cree identity, because representation matters. And I think we're starting to realize that more and more in science. And so I'll just, I'm reminded of George M. Johnson, who's a Black scholar. He's a Black writer, and he wrote a book called Not All Boys Are Blue. And he had a quote in there, something along the lines of, you know, sometimes you can't see yourself until you see other people like yourself just merely existing or living or thriving. And so that really kind of spoke to me, and the effect that your presence, whether just merely existing, can have a positive influence. And I want the sort of the people in attendance to recognize that you can have that influence on other people. So if there's parts of your identity that are worth sharing, I think you'd be very surprised at the very positive influence that can have. So I'm going to breeze through this slide because as I already summarized it very well, but this is just an overview of my academic timeline. And just to give you a bit of context, because we'll be talking about this throughout this talk. So that's sort of the introduction. Indigenous people are sort of at a disadvantage because there's a lot of protocol you have to do before you actually get to the talk. So here's one of those sort of beadwork, artwork metaphors. And this is how I want to discuss my presentation outline. So on the left, we have sort of a river or a road or a path, and it's meandering and it's not always clear where you're going. And that's sort of a metaphor for my career trajectory. And so we do end up getting where we're going. It's just sometimes you don't always know how you're going to get there. And this is sort of the conclusion on the right, which is these two overlapping discs representing, and sort of in my mind, Indigenous and Western worldviews. And as they overlap more, things come more into focus. And that's kind of what I'm hoping we'll get out of this, that we really, to do science well, we do need to incorporate everybody's worldview, particularly blending those Western and Indigenous perspectives. So my talk is sort of broken down into these 4 categories, sort of the PhD postdoc, sort of the current level I'm at, and sort of the future. And these are actually some beadwork that I've done. These are beaded cymes done in Métis style, as well as floral diagrams. And it sort of shows like how you don't see it at the time, but even when you're at your PhD level, you grow and you develop and you continue to develop. And that's sort of hopefully what we'll see as we proceed through this talk. So the PhD portion. You know, I can't see who's in attendance, so I can only assume that a lot of you have a bit of a botany background or a science background. So I might breeze through some of this just so we can get to some of the more Indigenous focused details further at the end. So just one second. There we go. So I'll start by saying that my sort of undergraduate education was highly focused on Western science, particularly tropical plant systematics and collections-based research. This was largely based at museums, which is a little unusual for an academic experience. So I was at the Museum of Nature and also as a predoctoral fellow for a brief visit at the National Museum of Natural History. So there's a lot of experience and overlap that many of the field museum folk can relate to, I believe. I'm just giving you a brief overview of the type of research that I was doing. And so this is the systematics of flowering plants, where you sequence DNA, build DNA alignment matrices, use similarities and differences to estimate phylogenies, and use that as an evolutionary framework to understand or develop, revised classifications, understand character evolution, address different taxonomic issues, understand biogeography, diversification patterns, etc. And so I did sort of the whole spread of this type of research. And so I'll just sort of walk through some of that now because it's sort of integral to my story of where I began and where I end up. So just mention that I did my PhD on the Fourbac family. This is one of the largest flowering plant families. It's extremely diverse. It's got very diverse growth forms, chemical diversity, morphology, just really, really interesting group, very complicated. And you might be familiar with some of the members, including here we have manioc, which is an important food crop. We have the rubber tree, castor bean, and some ornamentals, including poinsettia, crotons, crown of thorns, and the succulent cactus-like Euphorbia genus. So these are the sort of cactus analogs found in Africa and Asia. So despite all of this variation, they're united by an interesting trait, which is a 3-carpellate ovary that explosively dehisces and shoots its seeds out, and it leaves behind a persistent columella. So that's a really interesting trait that unites this whole diverse group. My research was on a subset of this this really diverse family called tribe Plucanishi. And this is an unusual group because they evolved a twining vine habit, as well as stinging hair defenses, and up here in the right corner, one genus that has this very specialized pseudanthial inflorescence. In addition to that, there's diverse pollen morphology, the evolution of edible oil-rich seeds, and a very interesting pantropical distribution. So there was a lot of very interesting evolutionary questions we could ask with this group. But of course, the problem back in the day was that there wasn't enough taxonomic sampling, DNA sampling. So a lot of that needed to be addressed before we could look into some of these more broader evolutionary questions, as well as taxonomy. So that was sort of the main focus of my dissertation, build that phylogeny and study its evolution. I'm going to go over just like very briefly the methodology. And so essential to all of this is reliance on physical specimens. And this is where herbaria, whether it's university or museum plant collections, were so essential to getting access to this diversity that, you know, you're in North America, you can't easily access it. And then if you don't have any samples and you have the funding, you could always go and collect it from living material, which I did quite a bit of. And so on one hand, while this is so essential to the type of work that I was doing, it's also where I found I had a lot of the most ethical conflicts. So we'll get into that in a little more detail. This was both like the bittersweet realization for why I sort of moved away from this type of work. In addition to that, this is just a very brief overview of the phylogenetic approaches. So we collect the data, sequence the DNA, build those matrices. You estimate phylogenies using different statistical programs, Bayesian, maximum likelihood approaches. And then from there, hopefully you get a strongly resolved tree, and you can use that as an evolutionary framework to study downstream analyses like biogeography, character evolution, and so forth. So this is like a very general workflow that I would apply to many of my studies. And so it's funny, in the interest of time, I really sort of cut back how much time I dedicated to this. So it almost feels like cheating to not go into all this detail, because normally this would be a major focus of my talk. But, you know, we're growing and we're moving beyond that into Indigenous science now. But I will highlight that there were some very interesting studies that I was able to do with this. We resolved some phylogenies of some economically important genera. We could address a lot of the sectional classification issues and have really strong understandings of this genus after the fact. We could demonstrate that there was a trend towards aperture reduction, so the loss of pores and sutures on the pollen in this clade, which is sort of an evolutionary, not a mystery, but like, you know, why are you doing that? Because it helps, you know, it's a very stable trait to have these pores for the pollen tube to germinate through. So what is happening here to cause that reduction? It's a very rare and unique trend happening here. So there's some future work to be done for people interested in pollen morphology for sure. And then lastly, we were able to examine overarching trends in seed size evolution, the different ecological drivers and movement between different life history strategies. This was a lot of the work that I was doing up until recently. So as had mentioned, I did quite a lot of field work, and it was extremely lucky. And also it was also partly why I ended up sort of doing less of it at the same time. So I was very fortunate to go and do species collecting in Madagascar, Postgraduation. But I found that, you know, the more I was doing this, the more I realized that I was sort of participating or contributing to what I felt was like very extractive science. This is when you fly in, you fly out, you gather your specimens, you have sort of a minimal relationship with local botanists. You might leave a collection, a duplicate at their national herbarium, but there was little accountability and sort of capacity sharing. And maybe some people are doing this a bit more, but I just felt the way I was doing this was not, it was something that I was unsettled by, and I didn't feel like I could continue further. So that was sort of the big revelation, because I could see the stuff that I was doing were a lot of the same issues that were happening to my community and my family. I grew up in a resource extraction town, and I saw the environmental destruction and the use and abuse of our Indigenous communities' knowledges. And so that was sort of my biggest conflict of interest, and sort of central to how I sort of changed or adapted my career. Sort of jumping ahead, but what underlies a lot of this is that European and Western science is sort of intertwined with colonialism. And I think we do a really bad job of recognizing that as modern scientists. They all sort of arose around the same time: the age of exploration, the scientific revolution, the colonial periods. And because of that, colonialism is just embedded into the structure of how we do science. And it's a default view that we don't recognize the biases that it introduces. And so that's sort of the underlying theme here. And so because of that, I sort of toned down my field work, focused more on my methods, and sort of changed directions from there. So we're now entering the postdoc phase. And this is, it's an exciting time for me, actually. So this is, if you've done a PhD or you've done grad school and you sort of finish, you sort of feel like a weight is lifted. You're not so bound or constrained to your subject anymore. And so I was really able to explore new methods, and in particular develop a strong Indigenous network, which was something that I realized I needed if I wanted to sort of engage more ethically with the type of work I was doing. So for this, it was at the Montreal Botanical Garden, which is an amazing, really inspiring place to work at. I'll just say that I love museums. Like I love the museum I was working at and everything, but the plants they're pressed, they're dried, they're resting. But living in a living garden is it sort of feeds the soul, especially in winter. So in terms of new methods, up until now, all of my work had been done with Sanger sequencing, which was sort of a gold standard at the time, but phylogenomics took hold, and I was feeling really quickly left in the dust. So this was a real good opportunity for me to pick up these new methods, particularly targeted hybrid capture, next generation sequencing, where you could gather, instead of a handful of genes, 5 to 10 genes, you can gather hundreds of genes, supposedly to build stronger, more robust phylogenies. But in my experience, it sort of just complicates the story sometimes with competing gene histories. But also the ability to add functional genes to study aspects of molecular evolution besides just phylogenetic history. So for this, I studied the Leguminosae, or the Phebesia family. This is another extremely huge, large, cosmopolitan, species-rich, ecologically diverse, morphologically diverse plant family. So it was sort of right up my alley. Most of you are probably familiar with sort of the temperate diversity, which is largely the Papilionoideae, which have the typical pea-shaped flowers. But I just want to draw your attention to all these other panels, which are related to the different subfamilies that are mostly tropical in their distribution. And you can see that the floral diversity is extremely diverse, and you might see this if you ever vacationed in the South and not realize this was in fact a legume. But one trait that does unite them all is, of course, the legume fruit type, which is very characteristic. So that's another neat fruit type that unites these different large plant families. So my postdoctoral research was on one of these smaller subfamilies that was recently delineated, called Cercidoideae. It's a really interesting group. It's got these bilobate leaves, and which is a little different from other legumes that are either three-lobed or pinnate, mostly. They're really diverse in growth form, but primarily differ between being small trees to these big, robust lianas. So some of you, if you've done work in the tropics, are familiar with the monkey ladder lianas or vines, which facilitate movement of mammals through the forest. And in addition to that, it's a really diverse group. They exist in many different biomes, so wet, dry, savanna, temperate. whereas many of the other subfamilies are sort of niche conserved. So they're only in the dry, only in the wet. And so it was a really interesting group to do some evolutionary studies on. I'm not going to spend too much time here, but just to say I want to recognize that I had my phylogenomic training at the University of Brussels with the lab of Olivier Hardy. And that was a really fascinating experience. I'm really thankful for the people who go out of their way to help support sort of young scholars get the education and training they need. And I hope to really pay that forward someday. So what we did here is we tested different bait sets. Legumes are unique in that, not unique, but they're lucky in that it's a huge family, lots of research groups working on them, and lots of different bait sets. So we tested a few out and we decided to move forward with the angiosperm 353, which any systematists out here might be familiar with. So this is sort of an awkward segue, but right around this time, I think you can all sort of remember the day that COVID hit you, not hit you, hopefully not personally sick, but the day you sort of went home and never returned. So the COVID-19 pandemic, of course, is extremely devastating. And, I put this technical difficulty sign to sort of try to make light of like very tragic scenarios. But this is extremely serious event, lots of trauma happening to people and communities. And it just sort of really disrupted everyone's life. And so that's sort of, this is when it happened, right during the middle of my postdoc. And thankfully, I got a job after that, but I'm going to present, things that we were going to do, but I've not quite fully recovered. And so if you're still feeling behind, I'm here with you. I'm here in solidarity with you. So some of the big questions we wanted to answer were how is biome evolution happening in this group? And biomes are something that's coming up again and again in my work. And so these are regions that are easy to distinguish, but very difficult to define if you've worked with them. These are plant communities, largely plant communities, united by climate variables, vegetation structure, and in some cases, key lineages, like savannas might be very defined by grasses and so forth. And I won't go into too much detail because this part of the project has since been picked up by one of our students, Charlotte Hagelston-Renshaw. She's been doing some amazing work with this, developing taxonomic species lists, mining species occurrence data online, and developing distribution maps to overlay on biome, sort of biome pixels and things like that to identify where things are occurring. The really, really cool work that's up and coming that maybe we'll just have to get her here to present someday. But the key focus here is that we wanted to look at how evolutionary shifts among biomes, particularly like wet to dry, how that influences plant evolution, and if we could use those sort of abrupt shifts and responses as a proxy to understand what might be happening to climate change, how plants might respond to climate change. So that's sort of the biome evolution side of things. But in addition to that, I'll just briefly say that biomes has sort of come up also in my undergrad, my PhD work. So this is an unpublished chapter of my dissertation. That's also one of the things I need to pick up. And basically, we had this hypothesis that traits were really, really significant in the tribe. The evolution of these unique characters like twining habits, stinging hairs, and the flowers would be associated with those diversification rate increases as is suggested in the literature. But what we found based on tree and trait-based evidence was that traits were not associated with diversification shifts. And instead, it was biomes, shifts from wet dominant to dry dominant biomes, presumably are driving diversification in this tribe. And so that's sort of where we have to sort of look deeper into demonstrating that more clearly. But again, so biomes are coming up, whether you expected them or not, to be very powerful drivers of plant diversification, which could have huge implications for climate change. In addition to this, I expanded into, again, sort of new methods, new questions, and wanted to understand a little bit more about functional genes. So if you recall, we're sequencing genes that are very good for estimating phylogenetic relationships, but now we could also very easily add in, a couple hundred genes associated that wouldn't be good at estimating phylogenies, but would be pretty good at, or very interesting to understand functional genes and traits. So we picked a few here associated with leaf morphology, floral morphology, and seed and oil content. And so we have that data. We had as a really nice team of undergraduates looking at this. And so we sort of have our path set forward, but we probably need to get some dedicated master's student to help carry that forward. But that's some interesting work coming down the line. How do these traits evolve in response to biome shifts is sort of the context we're interested in exploring. In addition to that, thinking again about new methods, we have also at the University of Montreal, a collaborator, Etienne Liberté, who does a lot of work with leaf spectroscopy. And so for people who aren't familiar with this, the sun emits bands of energy at different wavelengths, and some of that's in the visible, some of it is infrared, and so forth. When that energy comes into contact with, say, like a leaf surface, based on pigments and water content and anatomy and different factors like this, part of that energy is absorbed into the leaf and the other part's reflected. And this difference gives you a very unique spectral profile of a leaf. And it turns out that due to those very minute differences in pigments, leaf anatomy, etc., this can be like almost a unique signature to the health of the plant, the phylogenetic affinity of the plant, and you could use this for a lot of very interesting applications. So that's sort of the new techniques that we're bridging into my current research, thanks to this sort of opportune relationship being sort of in the same institution at the same time. In addition to that, this also relates to some of the remote sensing work that I integrate later on as well. So I'll just mention that. So in addition to that sort of leaf spectroscopy questions, particularly what we wanted to do was see if we can estimate through modeling of leaf spectra different leaf functional traits. And it turns out that some of this very interesting work by Sean Kothari et al., they have a nice bioRxiv preprint available. They essentially propose that you can model leaf functional traits based on dried leaf spectra, which is a really powerful technique. If you could further extend the use of herbarium specimens into estimating leaf functional traits, rather than having to be in the field at the right time with fresh material. So this is a really interesting avenue of research with spectroscopy that we're looking to move forward, and that's part of one of my first new grants as a PI. But again, our main plan was to see if we could estimate leaf functional traits in Cercidoides from herbarium specimens. But again, COVID happened and sort of put a lot of that work on pause. So I just want to take a moment to sort of loop back to the story about how to bridge in Indigenous science. And it was really during this time in my postdoc that I had the opportunity to sort of think about ways to do this. And so I took the sort of conscious effort to seek out Indigenous community and really see, carefully, cautiously, I don't want to just, burst in and take up space. So this was the opportunity that did it for me. This is the summer internship for Indigenous peoples in Genomics Canada. And it's a one-week intensive workshop that aims to build capacity and scientific capacity through training undergraduates, community members, and so forth. And so some of these aspects I understood, the Western science side of things like genomics and bioinformatics, but I was really interested to learn more about sort of Indigenous and decolonial methodologies and bioethics. And so I can't emphasize how significant this was to my career trajectory. If you're an Indigenous student, and possibly I am assuming many other minoritized students could feel this way, you feel like you're lacking a community in general, like that speaks to that part of you that you don't see represented. And when you find that community, it really changes your whole outlook, and the questions and feelings you have are suddenly validated. And so that's what this did for me. And so if you're in that position, I can only further encourage you to seek out that community that you're needing. So this is what happened through this week-long internship, and it's been the foundation for a lot of my work moving forward. I'll just mention that this project was on chronic wasting's disease. And so this is another sort of beaded project I did that sort of outlines, it's a prion disease, so a protein disease that when it's misshapen, it becomes infectious and forms plaques, causes sort of mad cow type disease, but in cervids specifically. So this was a nice way of communicating that through media that was culturally appropriate to my family. And I'll just mention that it's really, there's a lot of these sort of Indigenous specific opportunities available. So if there's any Indigenous students out in the crowd and they feel like maybe genomics isn't the one for them, there's lots of other specific programs like this available. And it's just a really interesting and exciting time to reach out and find those networks. So we're sort of entering the new PI phase, which in hindsight, it's only been a year. So you know, the first part of the presentation was really like several years of my career, but you see how things have, you will see how things have been rapidly changing and growing and evolving once you get to the stage of being able to sort of control a bit more what you do with your work. And so for this is talking about the sort of era I'm in right now, which is at Agriculture and Agri-Food Canada. Sort of summarized as an experience of developing a new lab, developing further research in leaf spectroscopy, and very exciting, the opportunity to really exercise Indigenous scientific research. So what's also really funny and unusual and weird about this time is it's during COVID, and it's like the Zoom era, the Teams era. There were a lot of lockdowns. I'm sure if you've started a new job during this time, you can relate to not being able to go into your institution and be with your colleagues, or never really being in that environment. So that's sort of where this is. So really unusual time, exciting and unusual all at the same time. So let's see. So key to setting up the new lab was really developing a virtual community support. And this was a nice way of exercising Indigenous sort of values and priorities in the way I run my laboratory. And so it's quite like many other laboratories where you have, you know, your full-time employees, your students, but it sort of expands beyond into a virtual community where we have community members, people who are associated with the type of work you're doing, but they might be isolated, they need those additional supports. And so being virtual had been a real nice opportunity to develop mutual support networks. And this is a really common approach I find in Indigenous laboratories. And there's quite several examples out there. And so what we have, of course, you have to set this all up, but you have all the various things that take time to set up, more time than you think, the Slack, Miro boards, Dropbox, GitHub, ways of recording the work you do, all the Zoom meetings. But anyway, it's been, despite all the work, it's been a really great way to exercise reciprocity and feedback and mutual support. It's not just a take, it's like we're here to help each other out. And that's been really interesting to exercise as a PI. I was lucky enough to have my first grant approved, which was good because, you know, if you don't get it approved, you know, the funding can be a little spotty sometimes. And so this explored the fundamentals of leaf spectroscopy. And I chose this because I thought it was, I see such strong potential here with spectroscopy, and with the background I have in plant biodiversity, I felt it was really a nice combination. And so what I wanted to do was really build on the work by Sean Cathari and all of his collaborators on understanding how dry leaf spectra change through time. And this is because fresh regular leaf spectra, as shown up here in the corner, that's like a fully hydrated leaf. When you desiccate something and put it in the herbarium, there's changes, the pigments decay, water is lost. So some of those signals change, but some of them are stable regardless of whether they're dry or wet. So what we want to do is identify regions that are stable and then also see the stability through time. And that's important because, as you know, plant collections in herbaria, they could be 10 years old, they could be 100 years old. And so we need to kind of understand the parameters of how we can use dried leaf spectra, if they're only good up to a certain point or if it's really context specific. So that's sort of what this major portion is. So we're going to sample phylogenetic and functional diversity just from our local arboretum here in Ottawa. As well, there's two other smaller projects on estimating functional leaf traits of dog strangling vine, which is an invasive plant in Ontario. And essentially, we want to see if we can estimate or identify the functional traits that are associated with differences in biocontrol efficacy. And then further is to understand if we can identify different populations of this wild mustard, and specifically if we can find the groups that have this transgenic herbicide resistance. So as an additional, maybe quicker way of identifying it rather than DNA sequencing. So, so underlying all of this is actually the need for a spectroradiometer, which is a very expensive piece of equipment. And so we've been lucky that the funding has been approved, but it's been a very long procurement process. So we actually couldn't get to any of this work over the summer, but we have it approved. It's coming for next year, so we'll hit the ground running. Oh, excuse me, sorry, I burst into all of your ears. But in the meantime, we can build on this other part of this project, which is understanding leaf microbiomes, how they're associated with leaf spectra. And so for this, we're collaborating with the Calbo group with Etienne La Liberté. They've already sampled the spectra and understand the functional traits. And what we want to do is see if we can identify major differences in leaf microbial community and how those are influencing the leaf spectra. And this is important because leaf microbial community is really intricately associated with forest ecosystem function. And so if we can sort of better elucidate these relationships and use spectroscopy to understand that ecosystem function, I think that's sort of the ultimate goal. So this work is being conducted by Jordan Wilson Morrison, who's a student, a bachelor student in my lab, and soon to be a master's student. And we're examining sort of 3 case study scenarios, understanding this across phylogenetic diversity, across a sort of a single individual through seasonal change, as well as different species found along a nitrogen gradient. So it's going to be, very preliminary, and it'll help us determine the next steps in our research questions. So let's see. I know we're coming up to about my time, so I'm going to try to rush through some of these. So then the other project is ECO2, and this is Environmental Change One Health Observatory. This is a very big interdepartmental collaboration headed by Dr. David Lapin. It's conducted in the South Nation Conservation area. And this is where we're working with different farm landowner peoples and basically trying to elucidate the impact and value of natural infrastructure on agroecosystems. And sort of this One Health approach is really to study this one system from as many different points of view as possible. So we have sort of public health, entomology, water quality, climate, microbiome, plant, like just everything you could think of. And then from there, we're trying to better understand these interactions. And so how I fit into this was with my experience with spectroscopy and interest in spectroscopy. We're hoping to make the plant portion related to hyperspectral remote sensing, which is the next logical step of this type of work that I'd be doing. So we couldn't go out because of COVID-19. There were a lot of work restrictions for being out in public. So because of that, we pivoted to an indoor activity for the summer. So we have a student, Dominic Wood, who's been doing a systematic literature review on ecosystem function for hyperspectral remote sensing. And this will sort of guide our work down the line. And this could be estimating productivity, creating maps such as this to inform conservation practice or monitor how your practice is influencing or responding on the landscape, as well as to identify or track and trace invasive species or diseases. So really, really interesting stuff. And that's very luckily been supported by a nice new burgeoning network called the Canadian Hyperspectral Working Group, which we are lucky to be a part of. So it's been a very, so sometimes the stars align and you get the type of help and support you need. So that's one of our projects. This is another one. So very, very briefly, in situ germplasm. So some of the sort of management at Agriculture Canada is interested in moving away from ex situ conservation, which is sort of germplasm, seed banks, and so forth, and help industry and conservation authorities, etc., become active partners in preserving in situ germplasm. So you record and monitor what's currently in the environment and ensure it sort of persists there. And this has been done quite actively in places like Europe, but it's not been done much in Canada. And I mentioned this because the location we want to do this in is part of South Nation conservation, but it's in a very unusual, like unique and powerful area. It's the Healing Place Indigenous Garden. So I'll just mention that this is sort of co-managed by multiple entities, but I want to draw attention to co-management from Anishinaabe and Haudenosaunee communities nearby that had once an overlapping territory here. And so part of it is a traditional, they wanted to plant traditional medicines and trees for ceremonial activities, like a place for reconciliation and healing. But other parts of this property are basically old hay fields that have been sort of open up for this type of conservation work. And it felt really unusual to me that we would have such an opportunity to be co-managing this place, but that we would be doing in situ work without in the sort of Indigenous governance input on there. So that's sort of this where we're hoping to move forward. This is all sort of very current work. I don't want to name too many names because you know, it might not go somewhere, but this feels very promising. And so it sort of brings me to some of the sort of key messages that I think some of you might be here for. And this is sort of, how do you approach that type of work? And so the main approach here is through co-production. And I'll just highlight some of this information, but you want to develop projects that center Indigenous community interests and needs, that community members are active partners in that project and not the subject, which was very unfortunately common and even still is common today. You want to make sure that the data created benefits the community and that the outputs are accessible to the community, and that it supports principles of Indigenous data sovereignty, so that the data you're collecting is still accessible, that they have control over it and sort of shape how it's used. And so this is, again, a real interesting opportunity to exercise Indigenous governance, co-develop a governance structure on sort of agro-ecosystem land management, and with particular emphasis on, for lack of a better word, genetic resources. And I say, for lack of a better word, because Indigenous peoples have a very different relationship with plants, and they see them as, many communities see them as relatives, and they have, you know, very formal ceremonial actions, say when you're harvesting them and things like this. So I think there's some of these ideas don't translate fully, but I think it's really powerful that we could start incorporating better ways of being in relationship with plants in this type of conservation. So that's some of the type of work that's coming up. And then I know I'm getting really close to overtime, so I'm going to really breeze through this. This is Indigenous Data Sovereignty of Biological Collections. This is another project that's sort of near and dear to my heart because it speaks to my past experience with biological collections. And so as you may realize, there's a lot of indigenous data housed in plant collections, particularly on the labels, including the names, food uses, medicinal uses, or cultural uses. And some people would even go so far to say that sometimes the plants themselves are even indigenous data. And so what's the potential issue here? So briefly, so the data, it could have been collected without consent or ethical review. If there was consent, it might not have been collective. Consent may have changed. The data could be culturally sensitive or inappropriate to share, as well as the data could be inaccurate. And so we're trying to approach and use this data, but there's a lot of concerns underlying it that we don't think about too deeply. And this is sort of what I want to bring attention to. And further to that, there's a big push right now to digitize collections and create open access databases. And so a lot of this information could be published online, probably is already, and that puts indigenous communities at harm for their data to be mined. There's a big push in the ethnobotanical community to do that very thing, to take indigenous data that they don't have any responsibility to the community for. So to address this, we have a very interesting collaboration with South Dakota State University. This is a project headed by Dr. Maribeth Latvis, who is a botanist, as well as Dr. Mark Freeland, an indigenous scholar. And basically we want to, and I'm participant in this as well, we want to co-develop indigenous data sovereignty governance protocols for the herbarium in South Dakota State with the local indigenous community. And so there's some very interesting work on this coming up. And we'd like to use this as sort of a model that might help other institutions consider how they might uphold their duty to protect indigenous communities and their data. So I'll leave it at that. So the future, let's see. I'll try to breeze through this. So this is basically where do I see my work going? So very briefly, the co-production of indigenous food systems, this is something that's coming down the line. If it's not already being done, you're going to see a lot more of it. There's a real attention to it, at least in Canada, to support indigenous aspirations in food systems, whether it's agriculture or different food type work. This is to support both traditional food systems as well as interest in modern infrastructure, environmental management, and in particular an expanded understanding of agriculture. So it's not just, the prairies, it's, underrepresented areas like boreal, coastal, and wetlands. And this is all supported to support indigenous driven goals for food sovereignty, security, economic opportunity, and so forth. So all of these images are examples of work that's sort of in progress. Communities have interest in doing research on this to assert their autonomy and authority. And these aren't formalized yet, but I think you'll be seeing a lot more of this coming forward, either from me or many other research groups. In addition, I think as my hyperspectral remote sensing work picks up, I think it's going to pivot towards indigenous environmental management, in particular, upholding or supporting indigenous aspirations or desires to exercise their traditional management practices. So if you might be aware that there were a lot of wildfires in the Northwest provinces like BC. And this is due to a lot of mismanagement, and indigenous peoples want to exercise traditional management protocols. And so what we would want to do is see if we can use hyperspectral remote sensing to help support and demonstrate the differences in efficacy between, say, indigenous and non-indigenous practices. And this could be very helpful for changing how we approach environmental management, especially for climate change mitigation. And then very briefly, I'll say that integrative plant systematics, the type of work I've been doing, I'm sort of in it and I don't see myself exiting it too soon. So there's a lot of international collaborations that will keep coming. Genomics is really picking up, as well as continued work in biome evolution. and including collections-based aspects such as the data sovereignty and the dry leaf spectra. So that's sort of where I see my lab going in the future. So it's a very interesting and exciting time for me. So I know we're running really low on time, so I'm sorry if we don't get to the questions. But in terms of sort of my journey through how to bridge in Indigenous science, it was sort of a journey to do better. So part of that included recognizing my role in perpetuating harm. And that was sort of difficult to come to terms with. But then moving beyond that, carefully finding Indigenous scientific community and network, sort of easing into that respectfully, sort of catching up with the Indigenous theory. So I could only recommend, there's numerous courses such as Stay in Canada or off courses at the University of Alberta on Indigenous technoscience, for example, that can give you a strong foundation for how to work in these areas. And then sort of moved into having that foundation and being able to actually co-produce research that centers community benefits and interests. And then sort of last but not least, an overview of the type of work that I did, my scientific trajectory, Western scientific. So I sort of started off with tropical plant systematics, moved into braiding in different methods, so particularly phylogenomics and spectroscopy. And this sort of co-occurred with sort of my real desire to incorporate Indigenous methods and worldviews. And now I think it's really moving towards co-production of Indigenous-driven research. So this is sort of one example, my example, of how you might bridge Indigenous science into your work and to show that, a lot of changes are possible. So, all right. Thank you so much, everyone. I know I'm really right on the hour. So I see there's a bit of questions and chat. So I'm going to pass it over to whoever is, I think Az or Matt.
Can I ask him to warn? This is wonderful. It is our honor to have hosted you here. We are going to keep this room open for questions for a few minutes. I can see that some of you have been putting questions into the Q&A. Please make sure that you're putting your questions in the Q&A so that we don't miss them. There are a few back in the chat, and I've been going through and kind of letting people know, drop them over in Q&A. So I am going to let Matt field question and answer, and then there is a small group discussion happening after this. Those of you who have indicated an interest in joining that, you have been sent the link, and Emily is on hand to send links to additional people. Emily, can you maybe unmute yourself? I'm not sure if you can unmute. Yes, you can. If you would like to jump in on the small group discussion afterwards as well, please direct message Emily for the link to the next session. So Matt, I'm going to let you handle Q&A. Thank you, everyone, and thank you, Dr. Cardinal-McTeague.
Thank you so much, everyone. And sorry I went over time. There was a lot to this story, and I know I have to sort of pare it down.
It was wonderful. Thank you. I really enjoyed hearing it and learned a lot, and I'm very appreciative. So thank you so much. And thank you very much to Az for suggesting you and inviting you. And yeah, we have some questions if you have a few minutes from folks. On the Q&A, I'm not sure if you can see this here, but the first one is from Brittany Thompson.
Okay.
Are you able to see those, or should I read them out?
I think I got it, yeah.
Okay.
Oh, good question. So, sorry, excuse me.
Can you watch?
So, yeah, I'll just read it out. So Brittany had mentioned, so how is your focus?
Did you watch your?
Sorry, how has your focus
sorry.
to less colonial practice been received by peers? That's the question for people who haven't seen the Q&A. It's a very good question. I would say it's been really, it's been COVID, so a lot of this work has been happening sort of outside of a lot of the academic scenarios where you would get a lot of that reaction. But I would say there's some hesitancy at first, but I think because of not a lack of understanding, but it's really disruptive just to sort of tell people like, oh, science is colonial, and that brings hard feelings and things like this. So I think it's a very difficult subject to broach, but in general, people have been very supportive. So I say mixed response. Hopefully that's sufficient. And let's see, another question from Sean. So based on your past experience working with DNA sequences, can you speak about sharing benefits when working with DNA data of species important to Indigenous groups? Very good question. It goes on to say, are there conflicts of data private owned by Native people versus data open to all? So this is a really good question, and it speaks to a lot of the competing interest between open access. And I would say there's a huge movement right now, even within the government, open data, sort of open by default, like data is going to be produced and be available to the public. Like that's sort of the stance. But I think, at least where I find myself in certain locations where I have a voice, is saying like, that's great, but like there's some cases where we have to respect Indigenous data sovereignty, and that's a core principle that's embedded also in the government relationship with Indigenous peoples. So we have to, I don't think it's like in some cases, Indigenous peoples might be fine with their data being shared. And if there are benefit sharing agreements in place, then that's okay. But the issue is a lot of that isn't set out at this time. And so you have to really be diligent in establishing those relationships and knowing what Indigenous peoples or communities would like to happen. And that takes a lot of work. And so I sort of answered something. I don't know if I answered your whole question, but I'll leave it there for now. And then, okay, quite a bit of questions. Let's see, Arlene. So you highlighted Western European science methodology with the history of colonialism is different in other ways of knowledge production and with specific goals in mind that sometimes do directly interfere, disrespect, or invalidate some communities. Okay, so how do you see the relationship between sometimes contradicting values that inform questions and methods used in the name of science? So I think I'm understanding this question. And it's hard when I can't really see the audience. But I sort of interpret this as, how do you deal with sort of these conflicting worldviews where sometimes Indigenous perspectives are in conflict with Western scientific perspectives? And I do have sort of slides and information on some approaches to that. So one is sort of the two-eyed seeing approach by Elder Albert Marshall, who's Mi'kmaq, and this is a framework where you might have two worldviews, in this case Western versus Mi'kmaq. They might be in competition with one another, but you sort of meet halfway on the ground of mutual respect, and you sort of look past the differences and try to find the commonalities. And I think that's the framework that most people are trying to take now when they're co-producing. So there could be some things that are in definite conflict, but the more important thing is to find those areas that you agree on. And so I think I'll leave that there. I'm just going to take a sip of water.
I can start reading the next one if that's useful.
Sure, that'll give me some time to think.
All right. This next one is from Weston Testo. He says, hello, Weston says, hello, thanks for a great talk. Just wondering if you have thoughts on ways that natural history museums, like the Field Museum in particular, can be leveraged to engage with and support early career Indigenous researchers and students. Clearly, these collections are problematic in various ways, but are there opportunities to use these institutions to support and promote Indigenous participation and success in biodiversity research that other academic institutions may not be designed to?
Thank you, Weston. That's an excellent question. I haven't thought about that in particular, but I think in general, let's say botany as a field, I think a really powerful approach is to really make space for Indigenous students and scholars, any age really, to have a place and a voice in these areas, and that can be difficult because people sort of have this assumption that Indigenous peoples don't engage with science or education, for sort of outdated reasons. But we're here to say, like, yeah, we've always been interested in plants. We've always been interested in biodiversity. And our lack of participation has probably been from discomfort and sort of being engaged in these colonial institutions. So I think making space for that, and I think with the sort of co-production on entertaining or developing Indigenous data sovereignty principles with collections, I think that could be a really, I don't want to suggest it and then put Indigenous students in potentially unhealthy environments, but I think if it's done well and their voices are respected, I think that could be really useful. Anyway, I'm running low on steam, so I'll leave that answer there. And we have, okay.
If you have time for a couple more, there's a few maybe shorter ones here.
Yeah.
But yeah, someone is Kilo Wilkes says, good talk. Are there any plants that struck you as particularly problematic regarding data? For example, a plant that Indigenous cultures would not want to share data on.
Yeah, that's a very good question. So it will really be context specific. And something that I didn't get too much into this talk is that each Indigenous community has their own worldview and experience with science. And so one plant, might not be problematic to a community to have the data shared, but another could be extremely sensitive information, plants that could only be picked by certain members of the community, or if there's any data recorded about it, could only be held by certain knowledge carriers. I think there's many examples of this, but they're going to be extremely context specific. And that's part of the issue with exercising Indigenous data sovereignty. We tend to have this sort of pan-Indigenous idea of at least Indigenous peoples in North America. I want to just be clear, my concept of Indigenous peoples is really global, and I think that came across, but at least in the North American context, we think that we can consult one community and develop guidelines for the whole continent, and that's not possible because there's differences between peoples and their relationships with these plants. And so yeah, that's the major barrier. Like, how do you engage with every community? But you got to start somewhere. And so that's sort of what we're hoping to do, at least in South Dakota. I'll leave it there.
All right. Couple more. One that just popped in was, do you know of any rules for de-identification of data pertaining to specific data types or Indigenous data?
I wish I could discuss this further or clarify. I'm just curious on what de-identification means. And so I'll just sort of speculate de-identification as maybe like maybe censoring. How do we sort of remove or protect that data? And basically, like, I think one of the biggest ways that what isn't known maybe to everyone is that there are certain collections that are not public, or there could be plants related to what could be identified as plants of concern because of their drug or psychoactive compounds. There could be major components of this collection in herbaria that's not public because they don't, they just don't want to create problems. And so it's it's in our structures to keep some things private. So that exists. And so what we could do is have a sort of general rule of, we have Indigenous data. We've never engaged with these communities. We're not going to digitize it and post it. We're just going to withdraw it, at least from public database, until we exercise that relationship building. So I think that's a good enough response. Oh, and I just saw Richard respond. So yes, I think that was I think that kind of answered it.
Great. I think, yeah, maybe we should wrap it up now, if that's all right with you, and head to the group discussion.
Sure. I'm sorry, I'm just.
Oh yeah, if you want to field these other two real quick, that's fine too.
Oh no, sorry, I was just skimming through the chat, which I didn't have a chance to. Just wanted to say hello to the people who said hi, and thank you so much for your nice comments. It would have been so nice to do this in person. I love that sort of after chat. So anyway, I look forward to meeting with everyone who's able to stay. So thank you so much again for this opportunity.
Thank you. It was really wonderful, and we've got a lot of congratulations in the chat, which I'm saving and will send along to you. All right. Thanks again, everyone. And next week, we'll meet again. Dr. Chin Lee from the Field Museum will be speaking. Her talk will be about ecological dynamics of a temperate biodiversity hotspot at 2 spatial scales. So we'll be sending out the email for that soon. And thanks again to Dr. Cardinal-McTeague. Thank you.
Thank you so much, everyone. Take care.